Hello, everyone, and welcome to the Ciliate Genomics Consortium. I'm Emily Wiley.
And I'm Doug Chalker.
We're going to briefly tell you about our model for a student-centered professional learning community that advances faculty scholarship through course-based research. We hope that many of the elements of this model are transferable.
So as scientists, we're quite familiar with the elements of a research experience. Those include the use of scientific practices, such as making hypotheses, which then lead to discoveries that are broadly relevant. Part of that's also having collaboration and iteration. Those are all things we want to share with our students so they can actually feel like they're actually scientists.
So we would all love to do research with students in courses, but there are big hurdles. One being coming up with actual new research questions that are doable in a classroom environment. Another, designing new curriculum that might need to change with every new project. These things take faculty time, and faculty already struggle with trying to balance the demands of teaching and research obligations. So a group of us have discovered that with careful planning, you can design research experiences that benefit faculty research programs so that everyone wins. And it's even better if projects can be collaborative within and across institutions, support faculty research goals with discoveries and new collaborations, and benefit the broader science community. We've built a consortium around a model organism, utilizing common techniques to get many students working on faculty research problems in ways that also meet learning goals for our courses. In this way, faculty bring authentic problems to students, student discoveries contribute to faculty scholarship and allow faculty to further explore and develop new ideas that they can bring back to students. So you get a rich iterative cycle. Our consortium is organized around exploring the function of approximately 27,000 genes in Tetrahymena thermophila. The Ciliate Genomics Consortium has engaged now thousands of students at about 38 institutions in aspects of gene function research as part of their courses. And the trick was to develop modular curriculum to adapt to a wide variety of different courses. Together, we now have a battery of technical modules that can be applied to a variety of research questions in different courses. Any faculty member can take any selection and apply them in their courses toward advancing their own research areas and meeting their course goals. For example, we have modules that can be done through bioinformatics, in intro courses or advanced courses, or wet lab components, such as looking at gene expression or tagging a protein with green fluorescent protein and doing localization studies. So in this way, faculty can take the module that works for them and meets their goals and integrate it into their courses. And what's more, they can serve as the basis of collaborations within or across disciplines and across institutions and between courses in a single institution. And these kinds of collaborations have advanced faculty scholarship in ways that even lead to publication. And I'll pass it to Doug to talk about a different model.
Well, so we can take either these as standalone modules to build in for your particular learning goals, or you can do as I've done, is taking various modules and build them into a full research experience in one semester, where students start with basically the bioinformatics of a predicted gene and take it all through the cell localization and expression analysis like we described. And at the end, they're ready to publish a full report on what they've actually discovered. Next. So we're really, in this class, engaging students in molecular cell biology in the classroom setting, but they're doing original research. They have ownership because they are the ones who are studying the gene that they've selected. But then they're linked to either my own interest or research of my collaborators, and we've even published peer-reviewed publications from with the students as authors from the work they have done. But not always do we are able to publish peer-reviewed papers based on what any one student does. And because to overcome that, what we've decided to do is actually to build a database where we can actually see students disseminate their actual data in real time. Basically, at the end of the semester, they put their reports up on something we call SuperDB, or Student/Unpublished Research Results Database. And at this point, it's basically like a full research paper that they write, and they can even download a PDF of that. We're not going to show you the actual reports, but we encourage you to go to SuperDB. But what this has allowed us to do is to get the research results that the students are making into the hands of the right target audience, and that's the larger research community. So here's one part of a SuperDB report, which basically shows the linkage to a tetrahymena gene that actually then links back to the tetrahymena genome database. Researchers who use that database then can actually see the reference of the student result, and that can link back to, and they can actually look at students' results. These aren't peer-reviewed, but they're available for the larger research community.
So in our model, students are at the very center. They're collaborating with faculty and also disseminating their discoveries to the broader research community. And in doing so, they're able to see themselves as part of the research community, and this helps develop their identities as scientists.
And we've really built this from Emily and I up to a large collection, a consortium of more than 26 faculty at various different institutions across the country. We've done this largely by offering training workshops and having collaborations, is sort of shown here, and really by helping faculty do their scholarship with students in the classroom, we've really made a rich collaborative environment. And we would just so in addition to Emily and I, we brought along early in the process, Eric Cole at St. Olaf, Josh Smith at Missouri State, and Naomi Stover at Bradley. Naomi created the SuperDB. And through these collaborations, the start of this collaboration, through the larger collaborations and funding from NSF and HHMI, we built the Ciliate Genomics Consortium. And if you're interested, we'd really love to talk to you.